continue a discussion the lectures of a clinical chemistry. So our lecture today it's about the lipid, how the lipid is transported or are transported. So if we want to know how the lipids are transported, because we know that the lipids comes from many sources, as you can notice from here. So the first one, the lipids that is come from the diet, it transported through the chylomicrons. So the chylomicrons is responsible for the transport of the lipids from the diet. If the lipid comes from the adipose tissue, and we know that the adipose tissue that is stored the fatty acid, so the serum albumin is responsible for the transfer of the fatty acid that comes from the adipose tissue. And we discussed before, the serum albumin is also responsible for the transport of the unconjugated bilirubin and the transferred it to the liver. It also the triacylglyceride that synthesized in the liver, that also transported by very low density lipoproteins. Okay, so the triacylglyceride that is synthesized by the liver, transported by the very low density lipoproteins. And now we talk about the cholesterol. The cholesterol that is comes from the liver, transported through the low density lipoproteins. But the cholesterol that is transported from the dead cells, transported by the high density lipoproteins. And that why is the high density lipoproteins lovely lipoproteins, I can say. It's a good lipoproteins, while the low density lipoproteins is the bad lipoproteins. Again, I'm talking here about how we transport these lipids. The lipids from the diet through the chylomicrons, fatty acid that comes from the adipose tissue through the serum albumin, triglyceride from the liver through the very low density lipoproteins, cholesterol that comes from the liver through the low density lipoproteins, cholesterol come from dead cells that come from the high density lipoproteins. And as you can see here, the I can see this photo is explain everything. So the very low density lipoproteins is responsible for transport of a triacylglyceride from the liver. And then we have also, as you see here, triglyceride to the cell. So it takes it from the liver and transported it to the cells. Okay, and the chylomicrons that is take the lipids from the diet, and both of them they are go to the sometimes they are just come from the enterocytes, as you can see here. Like, just follow the arrows. Why this? Because the diet is come from the lipid that is coming from the diet, so let's go to the enterocytes, which they are the intestine of the cells of the intestine, sorry, and then they are just transported through the chylomicrons, and maybe they are transported it to the liver. If we talk about the very low density lipoproteins, it take the triglyceride to the cells. Low density lipoproteins take the cholesterol from the liver and take it to the cells, but the high density lipoproteins take it from dead cells and back to the liver. Okay, then this diagram is we just explain it from the other sides. Liver releases very low density lipoproteins because they are lipoproteins, and we know that the most of the proteins they are synthesized from the liver. So the very low density lipoproteins comes from the liver, and they are responsible for taking the cholesterol from the liver. And not cholesterol, triacylglycerides from the liver. Chylomicrons derived from the enterocytes are shown entering the circulation. Triacylglyceride are distributed to the cells through the lipoprotein lipase enzyme. Low density lipoproteins deliver cholesterol to the cells, and uptake occurs via receptor mediated endocytosis marked by the asterisk. High density are transporting cholesterol from the cells to the liver, so that is lead to release cholesterol transport. Let's explain everything. So the first one is liver releases very low density lipoproteins, and we know that the very low density lipoproteins that take triacylglyceride from the liver to the cell, this is the responsibility of it. And we understand from this sentence that the very low density lipoproteins is synthesized by the liver. Then we go to the chylomicrons, which is responsible for taking the lipids from the diet, and the diet just go through the mouth and enter the GIT system, so it will be the last step for it is the enterocytes. So the chylomicrons, they are synthesized by the enterocytes, which they are the cells of the intestine, and then entering the circulation. Then we talk about the triacylglyceride, that they are distributed to the cells through the lipoprotein lipase enzyme. So that how it's distributed to the cells through the enzyme is called lipoprotein lipase enzyme. And then we go to the low density lipoproteins, which deliver the cholesterol to the cells. So it takes it from the liver and then deliver it to the cells. How they just this lipids enter to the cell through something is called endocytosis. And high density lipoproteins are transporting cholesterol from the cells to the liver. Okay, so the high density transporting the cholesterol from the cells and transported it to the liver. That's why the high density lipoproteins is better than the low density lipoproteins. So the high level of high density lipoproteins is a good sign, while the high level of the low density lipoproteins is a bad sign, because it transports the cholesterol from the liver to the cells, so that is not a good sign. Now we go to the low density lipoprotein structure. As you can see, it's a very complicated drawing that showing us what is the structure of the low density lipoproteins. Let's start with the phospholipids. As you can see here, everything green is the phospholipids. And as you can see here, the phospholipid form a monolayer on the surface of the low density lipoproteins, provide structure and the stability, make the particle amphithetic. And what we mean exactly by the amphithetic, that's mean it contains hydrophilic and hydrophobic regions. So the hydrophilic region, it will be outside, while the hydrophobic region, it will be inside. So that is present in the surface, like just how to deal with the other substances that most of them they are hydrophilic. So again, phospholipid that is composed of hydrophilic and hydrophobic region, which give us the amphithetic characters that give that just like form the monolayer on the surface of the low density lipoproteins. Then we go to the cholesterol esters. As you can see here, by the cholesterol esters, they are just present in the center of the low density lipoproteins, and they are just the core component of the low density lipoproteins, consist of the cholesterol bound to the fatty acid, and they are formed to the hydrophobic core and take the majority. So they are the major of the low density lipoproteins. They are just attached to the fatty acid. As you can see here, the yellow is the cholesterol ester that we are described here, and it's a core component of the low density lipoproteins, and just composed of the cholesterol that is bind to the fatty acid and form the hydrophobic regions of the low density lipoprotein. So it's composed of the phospholipid and then composed of the cholesterol esters. The other thing is the apolipoprotein P100. This is the name of it, apolipoprotein P100, which is, we can see it's the blue one. This protein warps around the low density lipoproteins, plays an important function role if the low density lipoproteins help stabilize it, recognize it as a receptors on the surface to facilitate the cholesterol binding of this cholesterol, because it transfer the cholesterol from the liver to the cell, so they transported it via receptor mediated endocytosis. So if we're talking about these because just we said phospholipids, cholesterol esters, and then we talk about the apolipoprotein P100 that is present also. It just like warped the structure of the low density lipoproteins. It has an important role. It's maybe the main part that is responsible for the function of the low density lipoproteins. It mediated the binding of the low density lipoproteins to the receptors of these cells through the receptor mediated endocytosis. So this is the apolipoprotein. And then we have unesterified cholesterol, because we have cholesterol esters, and in the contrast we can say unesterified cholesterol, and we can say it's a free cholesterol, which is a pink one, that is present on the surface of the particle and interacting with the surrounding environment and just making interaction with the other environment. So as you can see here, this is the low density lipoproteins, and which is composed of the first one is phospholipid, that is composed of the phospholipids, which as I said, that is composed of the hydrophilic and hydrophobic region. The hydrophilic, it will be on the surface, the hydrophobic, it will be inside the surface, so that is give the emphatic structure, which is known as the phospholipids. And then we talk about the cholesterol esters, which is form the core of it, which is composed of cholesterol attached to the fatty acid and form the core of the low density lipoproteins. In contrast, there is unesterified cholesterol, which we can say it's a free cholesterol, and that is present also on the surface of the cell. And the blue one, which is known as apolipoprotein P100, that is mediated the binding of the low density lipoproteins to the cell, so facilitate the transport of cholesterol from the liver to the cell. So this is the structure of the low density lipoproteins, because we're just cautious about how that is can be happen. As you can see here, the diameter of the low density lipoproteins here is about 22 nanometers, as you can see here, which is, we can say it's very small. So the diameter of the low density lipoproteins is 22 nanometers. Then, because I said the apolipoproteins P100 is responsible for this, which we can say receptor mediated endocytosis, how the cholesterol enters from this, because the low density lipoproteins transport the cholesterol from the liver to the cell. My question is how the low density lipoproteins transport this cholesterol to the cells. That's a question. So as you can see here, just want to show you in a very complicated drawing how that is can be happen, this endocytosis. As you can see here, this is the low density lipoproteins. It will be enter the cell. Okay, so here it composed of cholesterol esters. So here is the cholesterol esters, and here, which is known as the low density lipoproteins, because we know that the cholesterol esters is present in the core of it, and there is apolipoprotein P100 that is warped, we can say, around the low density lipoprotein. So what is happened? The first step is this step, low density lipoproteins binding. So the low density lipoproteins binding receptors that is present on the surface of the cells, forming complex with the cell membrane. So as I said, apolipoprotein P100 that is present on the surface of the low density lipoproteins, it just make a combinations or a complex between it and the membrane of the cell. So this is the first step. Then the second step is called internalization. Why we called it is internalization? Because the low density lipoproteins receptor complex is then brought into the cell. Okay, and then also the process is called endocytosis. The cell membrane warped around the low density lipoproteins, forming vesicles that is formed to the cell. So as you can see here, these lipoproteins receptors with the membrane, then it make just a complex. Then the membrane, it will surround around the lipoprotein and form this complex. Okay, these are, we can say, this receptor that is present in the membrane, and then this complex, it will be appear like a vesicle by the endocytosis. And then what is happen? Lysosomal hydrolysis. The vesicle fuses with the lysosomes. The acidic environment breaks down the low density lipoproteins into cholesterol and cholesterol esters. Okay, and cholesterol oleate amino acids. So as you can see here, then when it diffuses with the lysosomes, which is the three steps, then the lysosome, it will just, because of the acidic environment of the lysosomes, that is lead to the hydrolysis. So that is lead to breakdown of the lipoproteins. As you can see here, this is, we can say, amino acids, and these are the cholesterol oleate and the cholesterol 3 compounds, as you can see here. So this hydrolysis, that what is happen, that the amino acid, cholesterol oleate, and the cholesterol, and then the cholesterol, it will go inside these cells. And by this mechanism, that how the low density lipoproteins transport the cholesterol from the liver to the cells. I mean, just focus in the endocytosis. Again, how that is can be happen, the low density lipoproteins, they are just bound to the cell membrane and forming a complex. Then the cell membrane round or warped around the lipoprotein, forming a vesicle. Then the vesicle fuses with the lysosomes, and then it happened the hydrolysis because of the acidic environment of the lysosomes. That is lead to the breakdown of the cholesterol into amino acid, cholesterol oleate, and the cholesterol, which is so important is the cholesterol that is will be inside the cells. And that's what we call it low density lipoproteins receptor mediated endocytosis. Then I will take, or I can say, I will explain more how the lysosomal hydrolysis happen, which is the three steps or third steps that is occur here. So the lysosomal hydrolysis, the acidic environment breaks down the low density lipoproteins particle, releasing, as I can see, to cholesterol, cholesterol oleate, and amino acid. The cholesterol, which is so important for the cell membrane synthesis and for the synthesis of other molecules, as we can say that the cholesterol is also so important for the most structure of the cells. Actually, most of the structure that is present in the cells is composed because of the cholesterol. The cholesterol, oleate, and the other fatty acids, and also the amino acid that is come from where the amino acid come from the apolipoprotein P100, because it's the only protein that is present in the low density lipoproteins. Now we just make facts about the low density lipoproteins. The first one, that the low density lipoproteins is the causative core, the causative factor for the coronary vascular diseases. Is the most important cause of the heart diseases. Actually, that's what I said. It's a bad sign of the low, it's a bad sign really if there is an increased level of the low density lipoproteins. Then we talk about the ApoP100 is the main protein in the low density lipoproteins. Actually, when I just focus on the structure, it's the only protein that is present in the low density lipoproteins, and the most important one is apolipoprotein B100. The low density lipoproteins is removed from the cell correlations by the low density lipoproteins receptors. How the low density lipoproteins cholesterol concentrations are measured. Actually, how we can measure the low density lipoproteins cholesterol, we measured it by maybe by the direct method, which is more accurate, more precise, and we will explain what is a direct method, exactly what is it, or what is another formula is called Friedewald formula. So we have another method which is known as a Friedewald formula, which is a quick way to estimate the amount using, like, it can estimate the total cholesterol, it can estimate the high density lipoproteins, or it's not estimated, but it just we estimate what is the low density lipoproteins or what is the concentration of it through knowing what is the total cholesterol or high density lipoproteins or a triglyceride or a triacylglyceride acid. So these are what we call them as Friedewald formula. So how we measure the low density lipoproteins through the direct method, which is more accurate, more precise, or we can say it's a Friedewald formula. We will explain what is a Friedewald formula, but we actually using the total cholesterol, high density lipoproteins, triglyceride to know what is the concentration of the low density cholesterol. So as you can see here, this is the Friedewald formula. Again, it's called the Friedewald formula, and we want to measure what is the Friedewald formula. The low density lipoproteins cholesterol composed of total cholesterol minus high density low protein cholesterol and then minus a triglyceride divided by the 5. So as you can see here, to measure the low density lipoproteins, we should have the total cholesterol, we should know the high density lipoproteins cholesterol, and then we should know the triglyceride acid. As you can see here, the total cholesterol actually it composed, we know the total cholesterol, that's mean the whole cholesterol that is present in body. So it will be the total of, or the summation of high density lipoproteins, low density lipoproteins, triglyceride. It's assumption of all this stuff. So the total cholesterol, it will be like this. High density lipoproteins cholesterol, it's actually good cholesterol, which is common excess cholesterol from the circulation, and then the triglyceride, the fats that is present in the blood, and we will divide it by the 5. It's not totally accurate, but it's good, and this methods work only of the triglyceride is less than 400 milligram per deciliter. So if the triglyceride acid is more than 400 milligram per deciliters, then the measurement, then we can't use the Friedewald formula. So the Friedewald formula, we use it when the triglyceride acid concentration is less than 400 milligram per deciliter. So the calculation, or we can say the equation of it, equals total cholesterol minus high density lipoproteins cholesterol minus triglyceride divided by the 5. Then the other method, as we can say, it's a direct method, maybe homogeneous methods. We don't need manual separation or precipitations of the blood. Like we don't use the separation of the blood or precipitations of the blood. We use specific antibodies or enzymes that bind to the low density lipoproteins particles. Actually, we explained that before, how we can measure the high density lipoproteins, and we use actually offline treatment, online treatment. We have discussed that before. Actually, we use antibodies, sometimes we use also enzymes, like we said, cholesterol esterase, cholesterol oxidase, to measure the high density lipoproteins. low density lipoproteins. But these rules, as we can say, it's for high density lipoproteins, but here we are talking about the low density lipoproteins. Actually they are, it's like we make a general topic, we use a specific antibodies, we use specific enzymes, but I'm not now explaining what these antibodies and these enzymes to measure the low density lipoproteins, but it's a direct method. It's faster, more accurate, more convenient, because they are just formed in a non-fasting blood samples. It's more expensive. Examples for it is a direct low density lipoproteins assay, or APCAM low density lipoproteins cholesterol assay kit, or SCRIP research low density lipoproteins cholesterol assays. And there is another one to talk about. This is called HMGCO reductase inhibitors, used for the treatment of the low density lipoprotein cholesterol. But the question is how. So let's talk about how that this can maybe happen. As you can see here, we have the HMGCOA, which is 3-hydroxy 3-methyl glutaryl CoA, that's mean coenzyme A. So let's understand this, to understand what is happening, to know why that is lead to reduce the low density lipoproteins cholesterol. So as you can see from here, that the 3-hydroxy 3-methyl glutaryl coenzyme A and by the reductase enzyme, that is lead to the productions of the malveovate, which is a precursor in the biosynthesis of the cholesterol. Okay, and the examples or the enzymes that works by this is like a statin. Okay, and these, what we call them as a mevalonate, it's a precursor in the biosynthesis of the cholesterol. So when we just inhibit this enzyme, so the precursor in the biosynthesis of cholesterol will not be there. So that is a prevention the formation of the cholesterol. All the statin works by that. They just work by inhibition of the enzyme is called 3-hydroxy 3-methyl glutaryl coenzyme A reductase enzyme. And then we will talk about the high density lipoproteins. It's a good cholesterol that is lead to the reductions of coronary vascular diseases. AI is the main protein that is present in the high lipoproteins, because we said apolipoprotein P100 is the main protein of the low density lipoproteins, but ApoAI is the main protein that is present in the high density lipoproteins. How the high density lipoproteins reduces the risk of the coronary vascular disease is unknown, and that is like a chance for the research. How the high density lipoproteins measured by the homogeneous assay and also by measured in a fasting plasma sample, like supernatant after precipitation of the low density lipoproteins and the very low density lipoproteins, so we can measure it in the fasting plasma sample. Then we go to the triglycerides. Triglycerides are positively and independent associated with the coronary vascular disease. They are positively, that means they are increase the risk of the coronary vascular disease, and also independent, that means affect the coronary vascular disease and not affecting by other factors. Okay, do you understand that? Triglycerides are positively, that means it associated with the coronary vascular disease, but it's not as like when it's affected cardiovascular disease doesn't depend on the other factors. It's also an increase the risk of the pancreatitis, and we said that transported raw very low density lipoproteins, right? But in the case of the postprandial chylomicrons, because the chylomicrons transported it from the diet, the total glyceride can be measured in the plasma and the blood using enzyme assays also. Like sample collection, plasma or serum are separated, then we addition reagents like a lipase, which separate the triglyceride into the free fatty acid and the cholesterol. And the cholesterol, we use it to measure that, like we add another amount of the enzyme to measure the cholesterol, and the amount of the cholesterol will increases by, like the color intensity increases by the cholesterol, so we can measure by this way the triglyceride. So the triglyceride, it's 546 nanometers. The absorbance of the triglyceride, it's 546 nanometers. Good. Okay, because we said the lipid, it's from between 300 to the 700, and the triglyceride, the absorbance of it is 546 nanometers. So as you can see here, triglyceride plus H2O by the lipoprotein lipase enzyme, that is lead to the production of cholesterol and fatty acid, the cholesterol and ATP by cholesterol kinase, that is lead to the cholesterol 3-phosphate plus ATP. And then we do oxidase, that is lead to the dihydroxyacetone phosphate, and then the production of H2O2. And you know what is the results: H2O2 plus colorless compound by the enzyme is called peroxidase, that is lead to the colored compound, so we can detect the triglyceride by this mechanism. Lipoprotein lipase deficiency, that is lead to the production of increase the level of triglyceride, that is lead to the hypertriglyceridemia, and that is also lead to an increase the chylomicrons, and both of them, that is lead to the pancreatitis and acute coronary disease. So its a major cause is atherosclerosis, narrowing of the artery, so that is lead to the plaque disruption, thrombus formation, and if the obstruction is high graded, that is lead to the necrosis. So by this we just finish our lecture today. Thank you so much for listening. Hope you understand anything.